Welcome to Lecture 5.3, in which we will talk about enzyme structure, function, and regulation. These are the protein structures of amylase and catalase, two enzymes that we will be working with in the lab. Although we often represent enzymes with simple shapes and blobs, I wanted to show that like all proteins, enzymes have complex structures. Our learning objectives for this video are to understand why enzymes are important for life, describe their structure, and understand how they are regulated. Enzymes are defined as molecular catalysts, which means that they speed up metabolic reactions. You can usually recognize enzymes by their names, since most end in A's. Enzymes catalyze reactions by lowering their activation energy. Let's look at this energy curve, which shows how a chemical reaction would look without an enzyme in blue and with an enzyme in red. The addition of an enzyme decreases the activation energy, as seen by the smaller hump at the beginning of the reaction. As a reminder, the activation energy determines the speed of a reaction. Notice that the delta G, the change in free energy, does not change with the addition of an enzyme. The enzyme only affects the speed of a reaction, not whether or not the reaction is spontaneous. Since most chemical reactions in the body would proceed too slowly on their own for life, enzymes are critical for allowing metabolism to occur at the timescale of life. An important aspect of enzymes is that they can be reused over and over again. Since most enzymes are proteins, their shape is very important for their function. Like all proteins, they can become denatured at high temperatures and with changes in pH. And just to make this important point again, life is not possible without enzymes being around to facilitate metabolism. Here is a representation of an enzyme. The reactants that enzymes act on are called substrates, and when they interact, they form an enzyme-substrate complex. In general, a complex is a group of molecules that interact and bind together. The active site is the area on the enzyme that the substrate binds to. It's called the active site because that's where the enzymatic activity occurs. The amino acid sequence at the active site helps determine its shape, which in turn determines the substrate that the enzyme can bind to. That's why enzymes generally only bind to very specific substrates. Many enzymes also have another area called the allosteric site. This is an area where regulators bind. Regulators can change the overall shape of the enzyme such that the active site binds better or worse to its substrate. We now know that enzymes and substrates bind in an induced fit model. As mentioned, the shape of the active site is determined by the amino acids. However, the way amino acids are arranged can change depending on how they interact with other molecules. Here is an enzyme that is not yet interacting with its substrate. Its active site is not quite the correct shape for the substrate. When a substrate approaches the active site of an enzyme, it is actually the interaction between the substrate and the amino acids on the enzyme that makes the active site a perfect fit, as seen here when the enzyme-substrate complex forms. The enzyme then catalyzes the chemical reaction, converting the substrate into a product. The product is different from the substrate, so it will not interact with the active site in the same way. The amino acids at the active site will now change shape, pushing the product out of the active site. Now that the enzyme is by itself again, its shape returns to how it was before the substrate came along and is ready to bind to yet another substrate. This is how enzymes are able to be used over and over again. Enzymes can be regulated in multiple ways. As previously mentioned, enzyme activity is affected by temperature and pH. Its active site can also be physically blocked by something called a competitive inhibitor. Competitive inhibitors essentially directly compete with the substrate to bind at the active site. However, since they are not substrates, no reactions would occur, and the inhibitor does not change. Since it never changes, this inhibitor will just sit at the active site and block it until something kicks it out. There are also non-competitive inhibitors, which are inhibitors that instead bind at the allosteric site, changing the shape of the enzyme so that it cannot bind as well to the substrate. The allosteric site can also instead be bound by activators, which change the enzyme shape to better fit the substrate. Enzyme activity is also affected by the presence of cofactors and coenzymes, which are partners that the enzymes work with to perform metabolic reactions. Cofactors are often metallic ions. This image shows a close-up of glucokinase, which is something that uses ATP to add a phosphate group onto a glucose. This little green ball here is magnesium, and almost all enzymes that use ATP in some way need magnesium to help stabilize the reactions. A subset of allosteric inhibition is feedback inhibition. This is when, after a series of metabolic reactions, the final product goes back to inhibit one of the earlier enzymes. In this example, we have a substrate that binds to enzyme 1 to make molecule A. A is then the substrate for enzyme 2, which makes molecule B. B is the substrate for enzyme 3, which makes the final product. As the amount of this product builds up over time, it will start binding to the allosteric site of enzyme 1 in the pathway. This prevents the first substrate in the series of reactions from binding to enzyme 1, stopping the production of the final product. Feedback inhibition is often used to prevent the cell from making too much of a product and maintain balance. In this video, we learned that enzymes are crucial because they lower the activation energy of metabolic reactions, ensuring that they occur fast enough for life to be maintained. Enzymes act on substrates, which bind at the active site in an induced fit model and convert them into products. Allosteric regulators can bind at the allosteric site to change how well the enzyme binds to the substrate. Enzymes can be inhibited by competitive and non-competitive inhibition, as well as feedback inhibition. They can also be regulated by temperature, pH, and the presence of cofactors. We will be seeing a lot of different enzymes from this point forward, especially over the next 2 lectures, so make sure to understand them. Thanks for watching.